Microbes are everywhere. From walks around town with friends, to the deep endeavors of nature. Microbes are everywhere. From our shining moments on stage, through tractor rides and zip lines, microbes are always there. I wanted to research into that same thing that's always around through all our moments. Follow me through that journey. Now for the jarring question, what are microbes? Microbes are very small, and small living organisms that live everywhere but are blind to the naked eye. For my project, I collected samples from Lake Michigan and all over Evanston. In addition, I collected samples from Fox Lake in contrast to the Lake Michigan water, and sand, also other objects. To collect samples, we used gloves, inoculation loops, Sharpies, tubes, bags, and an agar plate, which you will see in the later clips. We started off by collecting samples from the soil at Hogan Hall and used a Sharpie to compare the size of the location. And then we collected samples from the elevator button and samples from the stair railing to see if there's any microbes on there as well. I then collected Fox Lake sand and Fox Lake water. I then collected a sample from a rock and placed it into a tube to observe later. I then collected some cool looking algae samples from Fox Lake to see what was in it. And then I used a scraper and a tube to collect samples from a tree at Fox Lake. Then we used this machine, which basically cleans air and makes it so that the areas in the box are areas with really clean air to make sure that the samples that we take aren't affected by any outside microbes. And then to sample the actual samples, we used an agar plate, which you can see in my hand right there, and then we used a pipette to collect a specific amount of water from the two lake water samples. We took the Fox Lake water and the Lake Michigan water, and then made sure to collect only the water from the samples and directly hit the water to make sure that there's no cross-contamination with the microbes from the outside. And then we placed a dot of that in the middle of the agar plate and then spread it using a specific spreading method. And as for the other samples, we used a sample of them using an inoculating tube and then switched it into water and used the same methods as we used for the lake water. And then we placed the samples into either a hot incubator box or place them into the fridge depending on the environment and climate of where we took the samples from. And we really didn't see any growth from the elevator button sample and from the stairwell sample, even after putting them in their respected location to grow. So we stopped really looking into those ones. I documented the growth of the microbes on the agar plate after being put in their desired environments, or like the heat box or the fridge, and some definitely grew more than others, such as the rock, which grew tremendously in color and size, and stuff that didn't really grow that much, like the tree. Now, this does not mean that there wasn't any microbes in the location. It could also mean that maybe the agar plate wasn't the desired growth environment for the certain microbes that were in that area and that we picked up. I also wanted to explore isolating some colonies, so I took a inoculation tube and I picked out the colony that I wanted to test out, and I did the pattern on the agar plate like you do, to make sure that everything's spaced out evenly and well. I wanted to pick out some that were similar in size and color, so I chose to focus on an orange kind of microbe that was found in the Fox Lake water and the Fox Lake rock, and see if there was any similarities. And looking closer at these colonies, they might look the same, but they actually weren't the same. Under the microscope, we could tell that they were completely different. They just had the same kind of red color. But something similar that they both had was the fact that they both smelled feral. And this is pretty common, especially since the area in which we collected this sample, which was Fox Lake, is known for having a really big garbage population around the lake and fish head carcasses due to the amount of fishing that is done at this lake. Then, in the words of Matt, we used a big fancy microscope to look at the different microbes and figure out exactly what we have on our guard plates and figure out the similarities and the differences between everything that we found. And we took a couple pictures as well. And thanks to the big fancy microscope, we were able to take a bunch of pictures of the different samples. And here we could see an amoeba-looking thing with a darker nucleus and some cocci, which are circular kind of figured microbes, and rods, which can be distinguished by the rod shape of them, as seen here. And I wanted to find out how many more microbes possibly Fox Lake had than Lake Michigan in the water. So I used the microscope to be able to take a picture of each sample to be able to count roughly the amount of cells. And then I took that number and multiplied it by the cover slip, which was 79.85 over the inoculating volume in milliliters. And from that, I was able to calculate the cells per milliliter that were in the water samples that we took. And after I was able to do all the math, I concluded that indeed Fox Lake had more microbes and cells per milliliter than Lake Michigan by a significant amount, almost double the amount of microbes that were in Lake Michigan. In conclusion, the research I did was really to explore the microbial communities all around me back in my hometown and all around Evanston. It's safe to say that there's a large abundance of microbes all around us, even if sometimes we can't grow them as well for them to be able to thrive in our labs. The process of being able to see, learn, and study these microbes is very precise. Microbial communities can be red, orange, yellow, and all sorts of other colors and carry all sorts of smells. They can be all different shapes and all different sizes, all uniquely their own. Microbes are truly a diverse thing that make it so much more intriguing to learn. Thank you.